Hi everyone. So let's go ahead and take a look at section 4 of chapter 22. So we're diving into the adaptive immunity here, and this part is just an introduction. So we're going to be taking a look at what antigens are. We're going to be looking at these receptors that are found on T lymphocytes and B lymphocytes. We'll talk about how antigen presentation occurs. So this is kind of setting the stage before we talk about the actual functions and what takes place in the adaptive immune response. So before we do that, I want to go in and just remind ourselves the differences between the innate and adaptive response. So let's take a look at this nice little website right here. So this is going in, it goes into a lot of detail. So if you're interested in it, I'll put the link up. But I just want to warn you that there's a lot of detail in all of these little sections that it goes through. So I just want to go in and look at the basics here, the differences between the innate and adaptive immune response. And again, we've gone through this, but we can kind of reiterate what is happening here. So this is going in and talking about the difference between the innate and adaptive immune response as far as components. We went through all of these physical and chemical barriers. We went through all of these different cells. We went through inflammation and fever. What we're going to be taking a look at now is the humoral immunity and how B cells are going to be responsible here in the adaptive immunity, and also cell-mediated immunity where T cells are going to be involved. Again, as far as activity, the innate immunity is always present, it's always there, but adaptive immunity, these T cells and B cells are normally just hanging out in the secondary lymphatic structures, and they're usually silent. They have to be exposed to an antigen first, and then that's when they are going to become active. So because of that, that's why the adaptive immune response or adaptive immunity is going to take a little bit longer to respond, so 1 to 2 weeks. But when it does respond, it is very potent. It's much more potent than the innate immunity. So yeah, innate immunity, it does have a lower potency, but it's really effective at fighting off some pathogens. But sometimes it's not effective enough, and that's why we have to have the adaptive immune response. Here's specificity. Remember that innate is also known as non-specific because it's very general as to the different pathogens that is going to respond to. So it's going to kill off anything that it can. It doesn't make any kind of fine distinctions like adaptive immunity will. These are adaptive immunities known as specific immunity because it will go in and target one single specific antigen. As far as course, again, innate, it attempts immediate destruction. If it can't, it will contain the infection, which is great. It will slow down growth until the adaptive immune response can come in and take over. As far as memory, again, I've mentioned this many times, no memory in the innate, but this is what we're going to have in the adaptive immune response is memory cells. They're going to remember these specific pathogens, so when we are exposed to it again, it is going to mount a much faster and much more potent second response. All right, so let's get back to our adaptive immunity here. So we're going to first start out by, it's talking again about what the adaptive immunity. So that kind of leads us, what we just talked about kind of leads us into this. We already talked about this. We already talked about this. Lymphocytes, T cells, and B cells are going to be the army that is involved here. So anytime that the lymphocytes and their products are released, this is collectively termed the immune response that is going to occur. So the adaptive immunity is going to be considered our third line of defense. Remember, first and second, we're in the innate immunity. So we have a couple of separate, or two branches. I don't want to say separate because of the fact that we're going to talk about how they work closely together. So I don't want to go in and say that these are separate, but they are going to be two branches that are going to involve two different classes of lymphocytes. However, I want you guys to know that these are going to be intermingled and very important and rely on one another. So we have the cell-mediated immune response or cell-mediated immunity. This is going to be where you have T cells They are going to be the main cells in this cell-mediated immunity. Another term for it can be cellular immunity, so you might see that as well. Cellular. Does that look like cellular? Hopefully. The T lymphocytes that are involved are cytotoxic T cells and helper T cells. And of course, we'll go through and talk about the functions. This is a main function of cytotoxic T, and this is going to be one of the big overall functions of helper T cells. But you guys will see more specifics on these later on. And here is the humoral immunity. This is also called the antibody mediated. It's not going to want me to write that over there. It looks really bad. Let me erase that. And try that again, right? Antibody mediated. That's much better. Antibody mediated immunity. As you can see, B cells are going to be the key players here. And we'll talk about how they differentiate and give rise to plasma cells. And then those plasma cells are going to produce antibodies. So the reason why it's called cell-mediated is because of the fact that these are very effective against antigens that are already within the cells, that have already infected a cell, or one that a phagocyte has taken in. This is known as humoral immunity because of the fact that they can respond to any antigen outside of the cell. It doesn't have to be taken into a cell and be presented. So that's one of the big differences between the two. So let's look at this word, this antigen, and what it is going to mean. So antigens, all pathogens are going to have something called antigens. So an antigen is going to be any substance that binds to a component of the adaptive immunity. And it really is these antigens that these T cells and B cells are looking for. So that's why pathogens are detected by lymphocytes, because of these antigens. So either an antibody or a T lymphocyte is going to go in and detect those antigens. So it's usually some kind of protein, or it's going to be some kind of large polysaccharide. But just know that each antigen is going to be unique for each infectious agent. So that's another key thing. So let's talk about what kind of antigens we have. There's the protein capsids of viruses, cell walls of the bacteria or fungi, bacterial toxins, tumor cells, all of these. These collectively here, because they are found on an infectious agent, are known as foreign antigens. So it's very important to talk about how our body, our own body cells, most of them are going to have self-antigens. And self-antigens are going to be these markers that are displayed on our cells to alert our body to let them know, hey, don't bind with me. I'm here. I'm supposed to be here. I'm supposed to be in Rebecca's body. I am a self-antigen. Those are our own body molecules. Foreign antigens are the ones that I had just mentioned earlier, the ones that you have the capsids of the viruses, the bacterial cell wall. All of those are going to be known as foreign antigens. And those are going to be ones that typically elicit some type of immune response from our T cells and B cells. So we'll talk about how T cells and B cells go through tests, and that is how our immune system is able to distinguish between self and non-self, is because they go through tests to make sure that they can do it. If they can't do it, they need to get gone, because the last thing we need is a T cell that recognizes a self-antigen as a foreign antigen. That's not good. That's how a lot of these autoimmune disorders are going to occur. It's because of this inability of a T cell, these lymphocytes, or something in the immune system to distinguish self from non-self. So again, it's because it could be from cross-reactivity, altered self-antigens, anything like that. So really good examples of autoimmune disorders is rheumatoid arthritis. This talks about rheumatic heart disease, but rheumatoid arthritis is one, type 1 diabetes. So with rheumatoid arthritis, you're going to have some immune system component that goes in and specifically attacks synovial membranes. with type 1 diabetes, you're going to have a component of the immune system that goes in and attacks the beta cells of the pancreas. So you see how it's going in and kind of attacking a very specific tissue because it's looking at this very specific self-antigen as foreign. Multiple sclerosis, it attacks the myelin sheaths of the neurons. There's Graves' disease. There's a lot of different autoimmune disorders. As far as what specifically our immune system is going in and recognizing, it is going in to look for this thing called an antigenic determinant, also known as epitope. So if we take a look at this nice little picture that we have over here, there's the antigen, and as you can see, it actually has several different antigenic determinants. Look, this one's shaped different, this one's shaped different, this one's shaped different. So these are going to be really specific sites on an antigen that some component of the immune system is going to recognize. So here's an antibody going in and recognizing this specific antigenic determinant. This specific antibody is going in and recognizing this one. So it's interesting because pathogens can have multiple determinants. There's an animation on that I have listed after this video if you want to take a look a little bit more on what antigen determinants are, epitopes. Look at it and watch that animation. It'll hopefully kind of clear things up for you. So any kind of antigen that induces an immune response, like this right here, let's say that this was just a random pathogen and this antibody goes in and binds to it, that antigen is known as an immunogen, and immunogenicity is going to be the ability to cause an immune response. So this is going to vary as far as it's going to increase the antigen's degree of immunogenicity by foreignness, size, complexity, quantity, all of that different stuff. Haptens are going to be these molecules that are way too small to function as an antigen alone. So usually they have to attach to some type of carrier molecule. So let's say that I have this nice little hapten molecule right here. Here's another hapten molecule. If this enters the body, not going to do anything. So usually what happens is they are going to use this carrier. Here's a carrier. There's my hapten. These are my hapten molecules. And they are going to attach to a carrier. Oh, didn't like me doing that one right there. Here it is attached to the carrier. This one, nope, didn't want me to do. There it goes. I don't know why it doesn't like me doing some shapes and sizes. But this thing, whenever those haptens go in and actually attach to a carrier, that causes it to be immunogenic. So it's kind of interesting. This is going to account for one of the hypersensitivities that you guys are going to look for, a type 4 hypersensitivity. So we'll look and discuss this whenever we get to it later on. All right, so moving on, this section is kind of a hodgepodge of different things. Again, it's just it's introducing some important components before we actually get to how the adaptive immunity works. So let's go ahead and take a look at the different types of lymphocytes we have, and we're going to take a look at their structure as well. So remember, T lymphocytes, cell-mediated immunity, and then there's our B lymphocytes, humoral immunity or antibody-mediated immunity. They are going to have very specific receptors. Remember, their responsibility is to go in and look for a very specific antigen. And how else are they going to recognize it unless they have a very specific receptor? So that's what we have here. Look at the little receptor. On the T cells, there's a TCR. It just stands for T cell receptor. And look at this on the B cell. It's a BCR, a B cell receptor. And look, it looks just like an antibody, doesn't it? That's because it actually is an antibody. So we'll get into that later on. Of course we will, right? So each of these cells, as you can see, is going to have about 100,000 receptors. And again, they are going to be very specific. You're going to have one helper T cell that's going to be able to identify one specific antigen. Then you'll have another helper T cell that is going to be able to identify another antigen. So you have millions of these you have millions of different types of T cells, and they are going to make sure that we don't, when we're exposed to different pathogens or just different antigens, with allergens and things like that, that hopefully we don't, elicit some kind of immune response. So that's, or if they do come into contact with an antigen that it has seen before, then it will elicit an immune response. We'll talk about how our body sometimes sees things as foreign when it normally shouldn't see things as foreign, like why people have food allergies and things like that. We'll take a look at that later on. So an interesting thing is whenever a T cell or a B cell comes into contact with an antigen, what they do with it is very different. So if we go back, this B cell can go in and just contact any antigen that is floating around in the bloodstream or the lymph. So this antigen can just be floating around alone. T cells are a little bit different. It's like they can't see the antigens and they need a little bit of help for them to see antigens. So their antigen has to be processed and it has to be presented to them in this nice fancy little way. So we'll kind of go through this whole antigen processing and things like that. And if we go back and look at this, I'm sure you're looking at this saying, oh, okay, there's the T cell receptor. Well, what is this little weird horn-looking thing over here? That's an additional receptor that they are going to have. Each of these are going to have. They're called co-receptors. And they are going to be very important in kind of this co-stimulation, making sure that the antigen is foreign so they can elicit some kind of attack. So look, here's the helper T cell, and look at that. Helper T cells have something called CD4 proteins. So this CD4 protein is a co-receptor. It's a class of co-receptor. So that's why sometimes helper T cells are also known as CD4 cells. The way that you can remember this CD4 goes along with helper T cell is because if you wrote helper T cell like this, look, helper T cell, doesn't that 4 look like an upside down H? I think it does. So that's how you can kind of remember. It doesn't look like it this way, right? But when I write it out like this, it definitely looks like an upside down H. So that's how you can go in and remember, okay, CD4 helper T cells. They have CD4 proteins, check. So we'll talk about how important they are, not only in assisting with the cell-mediated immunity, because it is a T lymphocyte, but also how it is going to be huge in humoral immunity. And it's also going to elicit some innate responses as well. So let's take a look at this cytotoxic T cell. Look, it is going to have a CD8 protein. So that's why sometimes cytotoxic T cells are CD8 cells. They're also called CD8 cells. How can you remember that? Well, look, if I went in and put an extra O down on these O's, because helper doesn't have an O, right? Only cytotoxic has an O. That looks like an 8. So that's how you can remember CD8 cytotoxic T cells. So again, they are going to be specific to these, and we'll also talk about how they are going to recognize different antigens that are presented on different complexes too. So again, we'll look at the different effects of cytotoxic T cells. They kind of tell you what they do in their name, how they're going to be toxic to chemical or toxic to cells. That's exactly what they are. We'll also talk about memory T cells. We'll briefly mention regulatory T cells as well, but these are going to be the major classes of T lymphocytes. Now remember I talked about how with T lymphocytes, they can't see the antigen on their own like B cells can. They have to be presented the antigens. So let's go through this whole process of how the antigen is presented. It has to be processed, and then it has to be displayed on these elaborate complexes. I guess they're not really elaborate, but these complexes are called major histocompatibility complexes, or MHCs. These are going to be these proteins, these transmembrane proteins that display the antigens for the T lymphocyte to actually see. So there's 2 categories of major histocompatibility complexes. You have MHC1s and MHC2s. So MHC1s are all going to be made and presented on all nucleated cells. So these are the ones, if it's a healthy cell, they're going in and presenting self-antigens. But as soon as that healthy nucleated cell is infected, then it's going to present a foreign antigen. We'll look at it in a second. So just remember your nucleated cells in the body, which most cells are nucleated, right? Except some of them we talked about. Red blood cells are not. So this is going to be your liver cell or a small intestine cell or your epidermal cells, anything like that. But you also have MHC2. So these are proteins that help display antigens, but they are going to be ones found on APCs. APCs are antigen-presenting cells. They do exactly what you think they do. They go in and engulf a pathogen, and then they are going to process that antigen and display it on an MHC to present to a T lymphocyte. The most common APCs are going to be dendritic cells. They are very good APCs, but also macrophages. And then we'll talk about how B lymphocytes work as APCs as well. So we'll get to these later on. So APCs sometimes will display MHC1s, but right now let's just remember it like this. We're not going to be taking a look at this one right now. We're going to focus on nucleated MHC1s. Again, we're not going to get into those little exceptions and things like that in this chapter, right? All right, so let's first take a look at how MHCs work and how they are going to display these antigens first on nucleated cells. All right, so you don't have to worry about the little specifics that's going on in here. What I want you guys to know is how they're going to be displayed. So let's kind of skip through this. I'll skip through this entire, I'm going to talk about it, but I'm going to talk about it very quickly. Look, here's some little peptide fragments of self-antigens. Look, it goes through the little rough ER. You see this weird kind of ant-looking thing? That's an MHC. So I don't know what it looks like to you. It might look like something else, some kind of other little critter, but a little ant-looking thing is an MHC. It's in here in the, it's made in the rough ER. And that's how it is going to combine with the antigen. You see how, look, it has it right there. It combines it right there. Pops it in a little vesicle, pops it over here to the Golgi, because the Golgi is going to, remember, process and package things that need to be either transported out or embedded in that plasma membrane. So here's our MHC1, and the reason I know it's MHC1 is because this is a nucleated cell right here. And look, it's going to be displayed. It has a self-antigen. This is what a healthy cell looks like. It's presenting it saying, hey, I'm self, please don't attack me. Let's take a look and see what happens when it's not healthy. Look, here's a virus. So one of those little capsid proteins are going to be embedded, or not embedded, sorry, it's going to be attached to an MHC1. How do I know it's an MHC1? Well, this right here is a nucleated cell, and it just got attacked by a virus. Again, it goes through the same process, but look, it's a foreign antigen. So this is really good. This is going to be displayed, and the lymphocytes that see it are going to be the cytotoxic T lymphocytes. They are the ones that are going to go in, respond to these foreign antigens on an MHC1, and go in and elicit an attack on this cell. So that's kind of interesting on what's happening there. Oh, I wanted to come back because look, the way that MHCs are, or how MHCs are determined, they're genetically determined. So you have one of these, I think it's on chromosome 6, that has over 200 genes on chromosome 6 are going to determine your MHCs. So they're genetically determined, which means that you are, of course, going to inherit it from your mom and your dad. So that's kind of interesting, right? And this is also going to be important when we talk about transplanted tissue as well, because these are going to be the things that display self-antigens, and it's really important for transplants. So now let's take a look at those APCs, how the antigen presenting cells are going to work. All right, so here is a bacteria, and look, here is a dendritic cell. It's going over here, or this could be, I mean, if you want this to be a macrophage, that's fine, but the picture has a dendritic cell. So it's going to go over here, and it's going to take in that bacteria. It's going to eat it. It goes through that whole phagolysosome, which goes in and breaks down the bacteria, and then eventually what's going to happen, look, here's the little MHC that's being processed. So it's processed a little bit different. Remember with the MHC ones, they were actually attached to the antigen in the rough ER, so it's a little bit different here, but you don't have to worry about the technicalities. As long as you know that this is an MHC2, who cares if there's 2 of those little ants in there, right? Don't have to worry about anything, any specifics like that. But just make sure you know that you have your little MHC2s that are binding a foreign antigen. Again, it's a foreign antigen. This is MHC2 that are displaying it. How do I know it's an MHC2? Because this is an APC. This is a dendritic cell. This is a macrophage or something like that. So that's how they're displayed. These are specifically going to be recognized by helper T cells. So just think 2 helper, or CD4s, right? One is going to be the cytotoxic T cells. So just think, C is going to be first in the alphabet before H, which is going to be after. So that's how you can kind of remember cytotoxic T cells. They recognize MHC1s. Helper T cells, they recognize MHC2s. So hopefully that helps you. Let's take a look and see what happens once they're displayed. Look at that. Look at this neat little dendritic cell here that's going in and displaying that. It has both of them on it. So again, let's just worry about dendritic cells having MHC2s right now. But as you can see, they can display both. However, we're going to worry about just one. So look, it's going in, and this T lymphocyte is going to recognize it. It's like, that is my specific antigen. And look, the receptor is going to be shape specific, and it's binding to the antigen that's on that class MH, the class 2 MHC. Look, here is your CD4 co-receptor that's going in and double checking it, making sure, yes, it is MHC class 2. Yes, it is the antigen that you are supposed to be attacking. So it's kind of a co-stimulation that's happening right here. So let's go ahead and take a look at organ transplants and why these MHC molecules are so important. So when an individual needs a new organ and needs a transplant, whether it's a kidney or a heart or a lung or anything like that, interesting fact, the only organ that doesn't have to be matched, that we're going to talk about here in a second, is the cornea. So that's kind of interesting. But any other organ, they have to be matched. And what I mean by matched is not only does the blood type have to match, but so do those MHCs. Specifically, what they look for are called HLAs. They're human leukocyte antigens. So they are going to be these MHCs specific to humans. Now, no 2 individuals are exactly the same except for twins, identical twins. So that's why if someone's in need of a either a blood, bone marrow transplant or any other kind of organ transplant, what they go in and do is they look for matches. They look for siblings. They look for parents. And then if they can't find matches there, then they're going to go to other places. So that's kind of interesting how it works. I'll put up a video that talks about HLA matching and stuff like that. You don't have to watch it, but if you're really interested in that, like with the Be Your Match organ donation, and if you're an organ donor or bone marrow donor, then it's kind of an interesting little thing that you go through. But what happens is that if they don't match, the natural killer cells or cytotoxic T cells, they actually look at these and they're looking at the antigens attached. If they're foreign antigens, or if these are going to be different, they're going to say, hey, this does not belong in this body, so I'm going to destroy this tissue. So that's why a lot of times right after a transplant, recipients are going to be given immunosuppressive drugs. I'm not sure, they might be given them before the transplant. Sorry, I haven't looked that up to see exactly when they start the drugs. But they'll be given immunosuppressive drugs to make sure that the immune system is suppressed, so those natural killer cells or those cytotoxic T cells or those immune system components aren't going to go in and attack these antigens that might be a little bit different. And that could be issues if you give an individual immunosuppressive drugs because their entire immune system is suppressed as well. So let's finish up this long video, right, by going in and taking a look at the overview of the life events of the lymphocytes. And this is kind of going to set the stage for us as far as what we're going to be looking at for the rest of the sections. We're going to be taking a look at how, let's look at the next slide. I should have combined these here. We're going to talk about how the T lymphocytes, how and where they are going to be formed. And we'll also take a look and see what does it mean about maturation of lymphocytes. So be able to recognize one specific antigen, but also be able to recognize them as foreign. So we'll go through that whole process. And then once they are presented, so once they're mature, we're going to talk about how they move to usually a secondary lymphatic structure, and then they just wait there for an antigen to arrive, and then we'll talk about how activation of lymphocytes is going to occur. And then once they're activated, what do they actually do to fight off that pathogen that they just found? So that is what we're going to be taking a look at in the next sections. So in the immediate next section, in section 5, we'll start off with this right here. So until next time.